Welcome to my course on microbial biotechnology. We are in module 1, which is basically discussing the introduction and principles of microbial biotechnology, classification and taxonomy of microbes. So, in this lecture number 2, we are going to study about the classification and taxonomy of microbes. So, this particular lecture is divided into the following sections. Section 1 starts with an introduction and gives an idea about the concepts of classification. Then the various type of classification systems which have been developed over time, like the Haeckel's 3 kingdom concept, then 4 kingdom classification, then 5 kingdom classification, and also it deals about the virus classification in brief and the 3 domain system. Then in section 2 we will be studying about the classification and phylogeny, and in section 3 we will be discussion about the particular domain called bacteria. Now to the beginner, taxonomy is the science of classifying, naming and identifying organisms based on shared characteristics and natural relationship. The word taxonomy actually is derived from the 2 Greek words, taxis, which means arrangement or division, and nomos, which means law. So, here we have certain guidelines or law which determines how the various microorganisms or living organisms should be arranged systematically. So, now when we tell taxonomy as a discipline, it has various aspects to it. The number one thing is classification. This involves the systematic grouping of organisms into categories based on shared characteristics. These categories can range from broad like kingdom to very specific like species. The second important thing in taxonomy is nomenclature. This is a system of naming organisms in a universally agreed manner. So, otherwise there will be confusion if we give multiple names, and these universal agreement ensure unambiguous usage of the same name by all individuals. The most widely used system is binomial nomenclature, which we will discuss in brief, where each species is given a 2-part name, the genus and the species, such as Homo sapiens for humans. The last important thing in taxonomy is identification. This is the process of determining the identity of an organism by comparing its characteristics with known organisms. Identification keys and databases are often used for this purpose. Now, let us start with classification and how the science of classification started. Carolus Linnaeus, who was an 18th century Swedish biologist, is often called as the father of taxonomy, and he classified living organisms based on their resemblances and similarities in form. So, humans appear similar to apes, and then you have cows appearing similar to buffaloes, cats appearing similar to tigers, and similarly you have certain plants which have stems and branches, and certain plants which do not have stems and branches. So, all these kind of morphological resemblances and similarities were considered by Carolus Linnaeus, and he divided the organisms into 2 main kingdoms, the Vegetalia and the Animalia. So, that was the simplest classification done by the pioneer of taxonomy. We know that microorganisms has evolved into different forms over time, and similarly the classification system has also evolved from time to time. For example, Haeckel put forward a 3 kingdom concept. The reason for his expansion of these particular classification was something related to microscopic unicellular organisms, which he has identified as well as other scientists of that time has identified, and they found that this did not fit into the 2 kingdom system designed for multicellular organisms. So, Haeckel revised this classification system put forwarded by Linnaeus. Haeckel was influenced by Darwin, and he developed the concept of the phylogenetic tree or tree of life, which illustrates the evolutionary relationship among different species. And in 1866, Haeckel proposed the tree kingdom concept, where he added a new kingdom called Protista, apart from the two proposed by Linnaeus earlier, the Animalia and Plantae. Haeckel proposed that organisms like algae, fungi and protozoa, which lacked tissue differentiation, should not be classified within the traditional plant and animal kingdoms. So, he introduced a third kingdom, Protista, to include these groups, which according to him are different from plant and animalia. Haeckel emphasized the evolutionary importance of single cell organisms, thereby highlighting their distinct evolutionary lineage separate from both plants and animals. So, in brief, Haeckel's three kingdom concept can be discussed as follows. As proposed by Linnaeus earlier, the kingdom of Animalia include all multicellular animals which are typically heterotrophic, that means they obtain food by consuming other organisms and characterized by complex structures and specialized tissues. Then comes the Plantae. These include all the multicellular plants which are autotrophic. They produce their own food through photosynthesis using sunlight, water and carbon dioxide, and they are also called as the primary producers, and higher animals survive on plants and or derived material and energy from plants for their survival. And the third kingdom that was added by Haeckel is the Protista, or which is also known as the Protoctista. This kingdom includes organisms with simpler structures compared to plants and animals which have complex and specialized tissues. So, these encompasses a variety of eukaryotic organisms that differ from animals or plants such as single cell protozoans and algae, along with some other simple multicellular forms. Now, as we have discussed about the influence Darwin had on Haeckel and his contribution to science by adding a third kingdom called Protista, as well as putting forward the phylogenetic tree or the tree of life, you can see here Haeckel's three kingdom concept, where you have Plantae and Animalia, and here it is the third kingdom, the Protista, and then you have various organisms in all the three kingdoms, and then they are classified into various branches depending on the level of their similarity in terms of morphology and other features which were used as keys for their classification. Now, building upon these three kingdom concept, particularly after the introduction of electron microscope in the 1950s, a new world was exposed to the scientist, and it was soon revealed that certain protists possessed a nucleus, and these were classified as eukaryotes, which are similar to plants and animals. Meanwhile, some other protists like bacteria lacked a membrane bound enclosed nucleus, and these were classified as prokaryotes. So, in 1956, Herbert F. Copeland proposed a four kingdom classification system that built on the ideas introduced by earlier biologists, starting from Linnaeus to others who have contributed, and he divided the kingdom Protista into two new kingdoms. The first new kingdom, Monera, included the prokaryotes. The second new kingdom, Protoctista, encompassed simple microscopic eukaryotic organisms such as algae, protozoa and fungi. Now, as I have already mentioned, that as organisms have evolved over time, so has their classification system. With the advent of newer technologies. For example, when the microscope become available, scientists could see that there are multicellular, unicellular organisms which are otherwise not visible to the naked eye. So, they included a third kingdom. Then later on, with the advent of the electron microscope, they could further see that there are some organisms within these microorganisms which have a defined nucleus. They included them into the eukaryotes, and some do not have that defined nucleus, and they kept included them as prokaryotes. Similarly, with the advent of another technology, Whittaker put forward a 5 kingdom classification. Here he particularly added fungi as the 5th kingdom due to the unique trait of external digestion before absorption. So, the metabolism of fungi was unknown earlier or not considered deeply. Whittaker considered this unique feature of these particular organisms which digest the food outside their body before they absorb it. So, he classified them into Monera, Protista, fungi, Plantae and Animalia. Monera includes all prokaryotic organisms like bacteria. Protista encompasses diverse eukaryotic organisms, microorganisms such as protozoa and algae. Then fungi covers multicellular eukaryotes with absorptive nutrition like moles, yeast and mushrooms. Then as usual, the Plantae consists of autotrophic multicellular plants, and Animalia contains all multicellular heterotrophic animals. One thing need to be remembered here, that these 5 kingdom classification of Whittaker did not or could not include the virus within its consideration. So, viruses are classified by the International Committee on Taxonomy of Viruses or ICTV, with classification reports published in archives of virology regularly. Virus classification uses Latin names for family, subfamily and genus ranks, but lacks formal categories higher than family and does not use binomial nomenclature for species. Here vernacular names remain popular among the virologists. These being altogether different subject, we will discuss about the classification of virus in module 2 in detail along with its structure and life cycle. Let us go back to Whittaker's 5 kingdom. So, here you can see the 5 kingdoms: Monera, then Protista, Plantae, fungi and Animalia. Now, these 5 kingdom concept, here organisms are classified hierarchically within each kingdom using the taxonomic ranks like phylum, class, order, family, genus and species. Each rank contains progressively broader groups of organisms. For example, species, this is the most specific level containing individual organisms. Genus includes multiple species, family contains multiple genera, so thus encompassing more species. So, as we move from species to kingdom, each taxon includes more diverse groups of organisms. Let us now discuss about the work by Carl Woese, who is a pioneering evolutionary biologist, and he fundamentally transformed the classification of life in the late 1970s through his analysis of small subunit ribosome RNA genes. The research by Carl Woese unveiled a previously unknown domain of life, which is known as archaea, and this shifted the paradigm from traditional morphology based classification to a genetic based approach. This breakthrough not only clarified the evolutionary relationship among organisms, but also had a profound impact on modern taxonomy. By 1990, it became clear that the kingdom Monera was composed of 2 distinct and unrelated groups, bacteria and Archaebacteria, further refining our understanding of the diversity of life. The 3 domain system. Carl Woese and his team at the University of Illinois Urbana-Champaign discovered significant differences in nucleotide sequences, cell wall composition, membrane lipids and antibiotic sensitivity, among archaeobacteria, bacteria and eukaryotes. They introduced the taxonomic rank of domain and proposed a 3-domain system dividing life into bacteria, archaea and eukarya based on ribosomal RNA sequences. The system clarified that archaea and bacteria are as distinct from each other as they are from eukarya, highlighting the fundamental evolutionary divergences. So, all these domains are considered to have a common ancestor from which they evolve, and this is known as the last universal common ancestor or LUCA. And from here you can see that the divergence first was into bacteria and the branch of archaea and eukarya. Then later on eukarya and archaea diverged during the course of evolution. Similarly, in eukarya we can see the emergence of microsporidia, then flagellates, animal, then fungi and plantae in the course of evolution. So, if you go into the bacterial domain, there also we can see various kinds of bacteria getting evolved over due course of time. We will be discussing some of these in the next few slides. So, now let us go to the second section, the classification and phylogeny. So, where we will be discussing about the basic taxonomic group and then the bacterial nomenclature, particularly ICNP and the binomial nomenclature. Now, classifying a microorganism require obtaining a pure culture which is a large uniform population of identical individuals. Traditional taxonomy methods involve examining the organism's phenotypic characteristics or the external characteristics which are observable properties resulting from its genotype. So, mostly we consider that genotype is a product phenotype is a product of the genotype and its interaction with the environment. Now, the various phenotypic characteristics that are considered in this entire process are morphological features, particularly the cell size, the cell shape, the arrangement, presence and arrangement of flagella, membrane and cellular layers. So, while discussing bacteria, we will be discussing all these morphological features one by one. Then other things like behavioral traits, particularly motility and responses to chemicals or lights, and then metabolic traits, the range of compounds that support growth, how these compounds are degraded and the resulting end products including the role of oxygen in these processes. Then another phenotypic characteristic is the cultural trait which results in the colony shape, size, optimal growth conditions, tolerance to oxygen and salt and spore formation in response to adverse conditions. Taxonomists group similar species into genera and related genera into families to reflect evolutionary or phylogenetic relationship, aiming to accurately represent the relationship among organisms. A notable example of this hierarchical classification is detailed in 9th edition of Burgess Manual of Determinative Bacteriology published in 1994. This authoritative system in prokaryotic taxonomy offers a comprehensive framework for classifying and understanding bacterial diversity. For those who are interested more in these taxonomic determination of bacteria, kindly please refer this book. Now, let us discuss about the classification and phylogeny. Taxonomic systems for biological organisms are structured hierarchically, as we have already mentioned. The highest level of classification is the kingdom or domain. The subsequent levels in descending order are phylum, class, order, family, genus, species and subspecies. To further distinguish a strain at the species level by unique characteristics, additional ranks below subspecies such as Petovar, Serovar or Biotype is also used. The basic taxonomic group in microbial taxonomy, the fundamental unit is the species. For higher organisms, a species is defined by interbreeding natural populations that are reproductively isolated from others. In prokaryotic taxonomy, a species is a group of strains with stable properties and significant differences from other groups. A prokaryotic species also has similar G plus C content and at least 70% DNA similarity based on DNA hybridization or sequencing data. So, briefly, the taxonomic hierarchy is as below, starting from the species. Below that we have of course the pathovars and strains, which we are not discussing here. So, species is a group of related isolates or strains. Genus is a collection of related species. Family is a collection of similar genera. Order is a collection of similar families. Class is a collection of similar orders. Phylum or division is a collection of similar classes. Kingdom is a collection of similar phyla or divisions. The number of different kingdoms varies depending on the classification system used. And finally, domain is a collection of similar kingdoms. Domain is a relatively new taxonomic category that reflects the characteristics of the cells that make up the organism. A strain is a distinguishable population within a specific taxonomic category. Strains within a species exhibit slight variations from one another. Biovars refer to variant prokaryotic strains distinguished by biochemical or physiological differences. Morphovars pertain to prokaryotic strains that differ in morphology. Serovars are prokaryotic strains that differ in serological or antigenic properties. We also sometimes use these as a classification tool. Biotypes refer to population or groups of individuals having the same genetic constitution. Example, two biotypes of the cholera bacterium, Vibrio cholera are Vibrio cholera classic and Vibrio cholera altor, and there are similarly many such examples. Now, let us learn about the bacterial nomenclature. So, bacterial nomenclature is governed by the International Code of Nomenclature of Prokaryotes or ICNP, which was previously known as International Code of Nomenclature of Bacteria, ICNB, and this is maintained by International Committee on Systematics of Prokaryotes or ICSP. So, those who are interested can visit this website and learn about the various activities of ICSP. Bacterial species validation is based on the approved lists of bacterial names published in the International Journal of Systematic and Evolutionary Microbiology, and this is a very important step in the whole nomenclature process. Now, let us discuss a little bit about binomial nomenclature, and we have already given some example in the past that, for example, humans have two parts to their scientific name, Homo sapiens. Basically, Carolus Linnaeus established the basic rules of taxonomic categories and developed the binomial system of nomenclature. And the binomial system involves naming organisms in two names. The first is the genus, capitalized, and the species in lower case, and the organism's names should be written in italics or underline, example Escherichia coli. The nomenclature is typically derived from Latin or Greek. For example, these name Escherichia coli are derived from these languages. Now, let us discuss about one of the important domains, the bacteria. So, here we will try to discuss about the size and classification and which is based on shape, flagella, and also gram staining. The domain bacteria. Antonie van Leeuwenhoek, a Dutch merchant, significantly advanced microbiology by observing bacteria in tooth plaque with a self constructed microscope, and published his findings in the Secrets of Nature discovered by Antonie van Leeuwenhoek in 1695. For this pioneering work, he is often called as the father of bacteriology. He referred to these microorganisms as tiny animalcules. The term bacteria, which means small stick, was introduced by Ehrenberg in 1829 to describe these tiny microorganisms. Bacteria are ubiquitous. They are found in soil, air and water. They are unicellular and prokaryotic microorganisms. Bacteria lack a true nucleus. Their genetic material is organized in a region called the nucleoid or genophore. Now, there is another nuclear material inside a bacterial cell. These are known as plasmids, which are small circular DNA molecules. These are separate from the nucleoid and they replicate independently. Bacteria have a thick rigid cell wall made of peptidoglycan, which provides shape and is located beneath external structures like capsules and flagella. This cell wall is distinct from the cellulose based walls of plants. Many bacteria are surrounded by a protective viscous capsule layer. In this figure you can see the structure of a generalized structure of a bacterial cell, where you have these bacterial flagellum and you have this nucleoid, and then apart from this nucleoid, which is a genetic material, you have another small genetic material here, which is the plasmid. Then you have the plasma membrane, the plasma wall and the capsule, which is kind of a protective viscous layer. Bacteria do not have chlorophyll, although some photosynthetic bacteria contain bacteriochlorophyll. They lack membrane bound organelles such as mitochondria, Golgi apparatus, plastids and endoplasmic reticulum. The protoplast includes the cytoplasmic membrane and the cell material enclosed by it. The cytoplasmic membrane, a lipid and protein layer, encloses the cytoplasm, which is about 80% water and contains nucleic acids, proteins, lipids, carbohydrates and ions. Unlike eukaryotic cells, bacterial cytoplasm lacks cytoplasmic streaming and a cytoskeleton. So, we already know that bacteria are very small and invisible to the naked eye, and if we compare the size of various kinds of bacteria with the weight of a human hair, which is roughly around 50 microns, we can see that cocci, which are round shaped or roughly around 1 micron or less than that, and then you have the rods over here and the spirochetes, which are different kinds of bacteria. So, this is a comparative size analysis of different kinds of bacteria when we compare to the human hair, which is visible to us. They are no doubt very small in size as an organism, but they are not all of the same size. So, bacteria have a wide range of sizes, and the average size of a bacterial cell measure between 1.2 and 2 microns in diameter to and 2 to 10 microns in length, and these actually vary from cell types to cell types. For example, the cocci typically have a diameter of around 0.5 to 2.5 microns, and there is an organism, of course, Begetua mirabilis. This can be as large as 16 to 45 microns in diameter and length up to around 18 micrometer. Similarly, there are bacilli or rod shaped bacteria. Their size ranges from 0.3 to 15 microns in length and 0.2 to 2 microns in diameter, and there are certain species called Dialister, Nemosyntes. These are as small as 0.15 to 0.31 microns. Now, we can divide or classify bacteria based on their cell shape, because bacterial cells exhibit a variety of shapes. Although bacterial classification was initially based on cell shape, this method is no longer the primary basis of classification, but still these are used for many practical purposes even today. Now based on their cell shape, the bacteria can be ellipsoidal, spherical or cocci-shaped. So, and then they can also have many variations like Coccus, Diplococcus, Streptococcus tetrad, Staphylococcus, Sarcini, and then they may be rod-shaped, which may be again Monobacillus, Diplobacillus, Streptobacillus and Pellicid, and they can be spiral-shaped like Vibrio, Spirillum, Spirochete, and there are other morphologies like filamentous bacteria, stalked bacteria, pleomorphic bacteria and budding bacteria. Now let us discuss these shapes one by one. Ellipsoidal, spherical or cocci. So you can see from this figure that they are spherical or globular, and you can very well compare their sizes with this scale, which stands for around 500 nanometer, and this is one species Staphylococcus aureus. This would have been in italics or it should have been underlined as per the binomial nomenclature. So, these ellipsoidal bacteria have a spherical or ball shaped cell structure with diameters ranging from around 0.5 micron to 1.25 micron. They exist in various forms including perfect spheres as well as oval, or they may be bean shaped and they may be slightly pointed variations. These cocci do not possess any flagella for cellular movement. These ellipsoidal bacteria exhibit specific arrangements based on their division patterns. For example, you can see cocci which is existing as single cell or single coccus, as you can see in this figure, and then they may be also appearing in pairs. So, we call them as Diplococcus. So, when the cells divide in one plane, they remain in pairs. We have many examples of such Diplococci like Neisseria gonorrhea, which causes gonorrhea, or Neisseria meningitidis, which causes bacterial meningitis. Then we have another arrangement called tetrad, where you can see 4 cells arranged in a plane. When the cells divide, it divides in 2 planes forming packets of 4. You have examples like Micrococcus luteus, which is an inhabitant of the skin. Then you have Micrococcus tetragenus. Then we have Streptococcus, which is basically a chain of cocci. If you look into it, these cells divide in one plane forming chains. So, you have Streptococcus pyrogenes or Streptococcus mutans, which are respectively responsible for strep throat or tooth decay. Another type is the Staphylococci. These cells divide in multiple planes in an irregular pattern, forming clusters resembling bunches of grapes. The examples of these type are the Staphylococcus aureus. It causes food poisoning, then toxic shock syndrome and also skin infections. Then we have Sarcini. Here cells divide in 3 planes in a regular plane, forming cuboidal or geometric packets, and the most important example here is Sarcinia lutea. Now let us discuss about the various forms of rod-shaped bacteria or bacillus. The term bacillus comes from the Greek word bacilli, which means rod or stick. So their ends can be rounded or flat or pointed, and their size ranges from 0.5 to 1.2 microns in diameter and 3 to 7 microns in length. They may have flagella, so they may be flagellated, or they may lack flagella, or they may be non-flagellated. Bacilli have a less varied arrangement as they divide solely in the transverse plane, which is perpendicular to the axis. Bacilli category includes many bacteria that causes diseases in plants. So let us examine some of the forms of rod-shaped bacteria, while certain species exist as a single rod, monobacillus. In many others, the cell remains attached after the cell division, leading to various arrangements. For example, in Diplococcus, they are present in Diplobacillus, like the Diplococcus, are present in pairs. Example, Diplobacillus pneumoniae. Then you have the Streptobacillus, which form chains. Example, Streptobacillus moniliformis. Now, this palisade is also a similar type of arrangement like the chain arrangement, but then here at one side a hinge is formed alternately. So, due to the alternate hinge formation, they form chains which are folded in space. So, Streptobacillus form chains, an example Streptobacillus moniliformis, and this causes rat bite fever. Palisade occurs very rarely, resulting in a palisade arrangement in which cells in a chain partially attached with a small hinge region at the ends, folding or snapping back upon each other, forming a row of cells side by side. Now, let us see the various forms of spiral sepate bacteria. Spiral sepate bacteria manifest in 3 different forms. The first one is the Vibrio, then you have Spirillum and Spirochete. Vibrio basically comma sepate, curved rods with diameters of around 1.5 to 1.7 microns and length up to 10 microns, and the best example is Vibrio cholera, which causes cholera. Then we have Spirillum, the plural for which is Spirilla. This is helical corkscrew sepate bacteria with a thick rigid cell wall and flagella. They typically measure 10 to 50 microns in length and 0.5 to 3 micrometer in diameter. We have examples like Spirillum minus and Spirillum volutans. Then we have Spirochete, which are thin flexible bacteria with endoflagella running along the length of the cell, allowing for flexible corkscrew like movements. An example are Treponema pallidum, which causes the disease syphilis. Now, if you compare between Spirilla and Spirochetes, we can see the overall appearance is rigid helix in Spirilla and it is flexible helix in Spirochetes. The mode of locomotion in Spirilla, there is a polar flagella, that is the flagella is in one side, enable the cells to swim by rotating like corkscrews, they do not flex. In Spirochetes, it contains periplasmic flagella located within seat and can swim by rotating or creeping on surfaces, they can flex. The number of helical turns in the Spirilla ranges from around 1 to 20, and in Spirochetes it ranges from 3 to 70, which is almost more than 3 times. And the Spirilla shows gram negative reaction as well as the Spirochetes also shows the gram negative reaction, and what is this gram negative reaction we will also discuss in one slide later. Then there are other morphologies in addition to all these morphologies that we have discussed. Some bacteria are spindle sepate or from branching filaments, while certain archaea display square or star-like morphologies. The exact reasons for these shapes are not fully understood, but they are believed to offer adaptive advantages. Some examples of various morphologies which do not fall into the earlier ones are filamentous bacteria, stalked bacteria, or pleomorphic bacteria. So, filamentous bacteria is a rod sepate bacilli growing in elongated chains covered by a tubular envelope. Filamentation is an abnormal growth of some bacteria that results in increase in the cell length due to longitudinal division multiple times without daughter cell separation, often without septation. The elongated cells contain multiple chromosomal copies growing in long thread-like strands. These are typically found in sewage water and effluent from sugar industries, and some of the examples are Sparotellus natals in water containing ferrous compounds, and then Leptotrix, Cladotrix, Nocardia and Begiatua. Stalk bacteria. Some bacteria forms thin extensions of the cell wall from the main cell body, which is a slightly hard appendicular structure and can help bacteria attach to surfaces and increase their surface area for nutrient absorption. Due to the presence of Prostaceae, they are called stalked or prostacate bacteria. Prostacate bacteria classified into two groups, bacteria where Prostaceae do not participate in reproduction, example Colobacter, and then bacteria where Prostaceae are involved in reproduction, example Hypomicrobium. Pleomorphic bacteria. Pleomorphic bacteria such as Acetobacter exhibit the ability to alter their shape and structure in response to changes in environment. These bacteria are found in diverse forms, demonstrating a range of morphological variations. Pleomorphism results from individual variations in cell wall structure due to nutritional or slight hereditary differences. Cornybacterium diphtheriaceae cells, typically rod shaped, shows diverse forms in culture such as club shaped, swollen, carved, filamentous and coccoid. Mycoplasma lacking cell walls exhibit extreme variations in shape, showcasing the pinnacle of pleomorphism. Classification of bacteria based on flagella. Based on the number and position of flagella attachments on their cell walls, bacteria can be categorized as monotrichous, lophotrichous, amphitrichous and peritrichous. What is monotrichous bacteria? They contain single flagellum at one end of the cell. Example is Cholera vibrio and Pseudomonas citrae. Lophotrichous bacteria contains two or more flagella, again at one end of the cell or sometimes in both ends of the cells, example Spirillum ildula. Amphotrichous bacteria contains one or many flagella positioned at the end of the cell, example Spirilla and Nitrosomonas. Then there is peritrichous bacteria which contains many flagella all around the cell, example Salmonella species and Clostridium species. Let us now discuss about one of the most important method of classification of bacteria, which is still used particularly in the clinics as well as in the fundamental basic microbiology research. So, this is known as the Gram staining or the classification of bacteria based on Gram staining. This method was developed by Hans Christian Gram in 1804 and it is a fundamental technique in microbiology for differentiating bacteria based on their cell wall composition. So, here you can see a slide where Gram staining has been done, which is of a pure culture of Micrococcus species, and you can see here cluster of from 4 gram positive tetrads in many places. Okay. Now, how this Gram staining is done? So, here basically a bacterial smear is fixed into a slide and then stained with crystal violet and then treated with iodine to form a crystal violet iodine complex. This is subsequently washed with alcohol or acetone. Bacteria that lose the stain quickly during the washing step are classified as Gram negative, while those that retain the violet color are classified as Gram positive. Now, why there is a difference in the retention of the dye? This dye retention is due to the structural differences in the bacterial cell walls. For example, the Gram positive bacteria have a thicker peptidoglycan layer that retains the stain. You can see here the thickness of the peptidoglycan layer, which is quite big in the gram positive, while this is very small in the gram negative. So, gram negative bacteria have a thinner peptidoglycan layer and an outer membrane, as you can see over here, that does not retain the stain. So, it is basically the peptidoglycan layer which is thick in the gram positive, which retains this dye. So, if you look into these diagram once more, you can see it lacks an outer membrane and have a thick cell wall composed of highly cross linked peptidoglycan, which retains the di-iodine complex even after alcohol or acetone washing. Examples of gram positive bacteria are Staphylococcus aureus, Bacillus anthracis, Listeria monocytogenes. And if you look into the cell envelope of gram negative bacteria, we can see that it basically has 3 distinct layers: the cytoplasmic membrane, a thin peptidoglycan cell wall, and an asymmetric outer membrane composed of phospholipids in the inner leaflet and lipopolysaccharides in the outer leaflet. They appear pink or red after counter staining with safranin because their peptidoglycan layer does not retain the di-iodine complex and is disrupted by alcohol or acetone. The examples of gram negative bacteria are Escherichia coli, which is also a model organism, and Vibrio cholera, which causes cholera disease, and Helicobacter pylori. So, with these we come to end of lecture number 2, which is basically the taxonomy and classification, and here we have discussed at length about the domain bacteria. We will discuss about the other domains in some of the future classes. Thank you.